Have you ever actually stopped to think about what's going on inside your body right at this very second? And I don't mean your heartbeat or your breathing, I'm talking about the absolute smallest level. If you think back to middle school biology, you probably picture a human cell as this sort of static, gelatinous blob with a couple of colorful shapes floating around inside. Well, get ready, because we're going to completely shatter that basic definition today. Welcome to This Explainer. We're taking a deep dive into the human cell's staggering, automated, city-like complexity. We are going to explore the microscopic metropolis that literally dictates our very existence.
Okay, let's dive right into this. Our source material challenges us directly with this idea. It tells us we need to look past those simple textbook definitions and really examine the molecular architecture, the specific biochemical pathways, and the structural subcompartments. And when we actually do that, we realize the human cell is definitely not a static blob. It's a highly advanced, bustling microscopic metropolis. We're talking about a city complete with its own command centers, manufacturing hubs, power plants, and even sanitation departments. To make sure we don't get lost in this massive microscopic city, here is our blueprint for today. We'll be touring 5 distinct districts. First, the Genetic Command Center. Second, the Translation and Transport Hub. Third, the Bioenergetic Engine. Fourth, Vesicular Waste and Metabolism. And finally, the Structural Architecture. Let's start our tour right at the very center of it all. District 1, the Genetic Command Center.
Think of this central district as City Hall. The nucleus is this highly protected, double-membrane vault equipped with massive, multi-protein security channels called nuclear pore complexes. They strictly regulate everything coming in and out based on an energy gradient. Now, what's incredibly fascinating here is how your actual genetic code is stored inside that heavily guarded vault. Your DNA isn't just loosely floating around in some tangled mess. It's beautifully and dynamically wrapped around these protein cores called histones. The cell actually organizes this into 2 distinct states. On one hand, you have heterochromatin. This is dense, tightly packed, inactive DNA, usually pushed to the edges. It's basically locked down in storage. But on the other hand, you have euchromatin. This is loosely packed, active DNA that RNA enzymes are actively reading to issue new blueprints to the rest of the cell. Still inside City Hall, we find a highly specialized subcompartment called the nucleolus. This doesn't even have its own membrane. It's essentially the factory that builds the machinery for the rest of the city. It's broken down into 3 functional zones. First, in the fibrillar center, RNA genes get transcribed. Then, in the dense fibrillar component, that RNA is processed. And finally, in the granular component, those processed strands are assembled into 40S and 60S ribosomal subunits. Once those parts are built, they're immediately exported out of the nucleus and into the cytoplasm, ready to get to work.
And that perfectly brings us to our second district, the Translation and Transport Hub.
So, we're leaving the quiet command center and entering the bustling, high-energy manufacturing district. Remember those ribosomal subunits that just shipped out of the nucleus? Well, when they join together in the cytoplasm, they form an 80S ribosome. These are incredibly complex ribosomes, which means the actual heavy lifting of linking amino acids together is performed by RNA, not a protein. And this brilliantly illustrates the rapid-fire assembly line they create. During translation, the ribosome forms 3 active pockets. First, the A site accepts the incoming amino acid. Second, the P site holds onto the growing peptide chain. And third, the X site serves as the exit, releasing the empty tRNA back into the wild. It's a rapid 1-2-3 process.
Now, if a ribosome is building a protein that's meant for export, a signal particle actually halts the process and docks the whole ribosome onto the rough endoplasmic reticulum, or RER. The growing protein gets pushed straight through a channel called the Sec61 translocon, right into the inner lumen of the ER. Once it's inside, that raw protein undergoes some really strict step-by-step processing. First, the signal peptide is cleaved off. Next, a 14-sugar chain is attached in a process called N-linked glycosation. Then, an enzyme called PDI forms the sulfide bonds to lock in its 3D structure. Finally, chaperone proteins swoop in for quality control to help it fold perfectly. If it misfolds, there's no second chance. It's immediately tagged and sent to the cellular shredder via the highly automated ERAD pathway.
While the rough ER is made of these flat sheets covered in ribosomes, it smoothly transitions into the tubular, interconnected network of the smooth ER. The smooth ER doesn't manufacture proteins at all. Instead, it's a massive chemical processing plant. It synthesizes lipids and steroid hormones. It also handles detoxification, like in your liver cells, where it uses the cytochrome P450 system to make dangerous toxins water-soluble so your body can flush them out. And it acts as a massive calcium storage vault.
In your muscle cells, the rapid release of this stored calcium is literally what triggers every single muscle contraction you make. Once those proteins are folded and the lipids are made, they get shipped in little cellular trucks, called vesicles, over to the Golgi apparatus. You can think of the Golgi as the microscopic shipping and sorting department. It's a highly polarized, strict processing line. Cargo arrives at the receiving dock, known as the cis-Golgi network. It then moves into the medial cisternae, where complex carbohydrates are remodeled and tweaked. Finally, it reaches the exit face, the trans-Golgi network. Here, the cargo is sorted, packaged into specialized clathrin-coated vesicles, and shipped out to its final destination in the cell.
Obviously, running a massive manufacturing and shipping district takes an astronomical amount of energy. So, for District 3, we are heading straight to the power plant, the bioenergetic engine.
What is absolutely mind-blowing about mitochondria is that they are evolutionary descendants of ancient bacteria. They even retain their own double membrane structure and duplicate all by themselves. The outer membrane is smooth, but the inner mitochondrial membrane is highly impermeable. So the crucial point is how this inner membrane folds over and over into sheets called cristae. This folding exponentially maximizes the surface area. And why do we need all that surface area? To pack in as many electron transport chain complexes and ATP synthase molecules as structurally possible. ATP synthase are literal rotational nanomotors. They spin at unbelievable speeds, driven by a proton motive force, just churning out ATP, which is the fundamental energy currency of your entire body. Deep inside all those folds is a gel-like fluid called the matrix. The matrix houses the enzymes for the famous Krebs cycle, it breaks down fatty acids, and amazingly, it contains its own separate circular maternal DNA. But mitochondria aren't just about giving life. They also hold the keys to cellular death. The space between the inner and outer membranes hoards a protein called cytochrome C. If the mitochondria becomes severely damaged and that cytochrome C leaks out, it triggers a rapid, irreversible self-destruct sequence known as apoptosis. Naturally, with all this intense energy production and manufacturing, a city is going to produce waste. Let's move to District 4, vesicle waste and metabolism.
Lysosomes are basically the cell's autophagic recycling centers. They are packed with a cocktail of destructive enzymes ready to break down old cellular components or destroy external threats. But consider this specific number, 4.5 to 5.0. That is the highly acidic internal pH of a lysosome, and it's actively maintained by proton pumps. This is such a brilliant safety mechanism. These destructive enzymes only function at that low acidic pH. So if a lysosome were to accidentally rupture, its enzymes would immediately become completely inactive in the neutral pH of the cell's cytoplasm. That prevents the cell from accidentally digesting itself from the inside out.
Working right alongside lysosomes, we have peroxisomes. These are essentially the intense chemical incinerators of the cell. They use molecular oxygen to strip hydrogen from very long-chain fatty acids. Now, that process naturally generates hydrogen peroxide, which is incredibly dangerous and reactive. But the cell is prepared for this. Peroxisomes contain massive quantities of the enzyme catalase to instantly neutralize that toxic peroxide into harmless water and oxygen. Without peroxisomes, our cells would just succumb to toxic stress, and we literally wouldn't be able to synthesize the vital components needed to build the myelin sheets that protect our brain nerves.
Finally, we've reached our fifth and final district, the structural architecture.
So how does this entire city hold its shape and manage transit without collapsing? The infrastructure holding this human cell together falls into three main structural classes. First, we have microfilaments, made of actin. These are super thin, about 7 nanometers wide, and they're responsible for muscle contraction and actually allowing cells to crawl. Next, we have intermediate filaments, made of proteins like keratins and lamins. These act like microscopic steel cables. They provide serious tensile strength and anchor major structures like the nucleus in place. And finally, we have microtubules. These are massive 25 nanometer tubes that act as the city's highway system. Specialized motor proteins called kinesins and dyneins literally walk along these microtubule highways, carrying massive organelles and cargo right to their exact destinations.
But where do these highways actually come from? They originate from the centrosome, which is the city's main microtubule organizing center. Sitting right next to the nucleus, the centrosome contains 2 centrioles structured like a 9-triplet wheel. Surrounding them are dense rings called gamma-TURC, which acts as the starting blocks for these microtubule highways to grow outward toward the edge of the cell.
And when it's time for the cell to divide during mitosis, the centrosome dramatically duplicates, migrates to opposite sides of the cell, and uses these very microtubule cables to literally pull the duplicated chromosomes apart.
We started today by tossing out that simple textbook image of a cellular blob. What we found instead is an awe-inspiring automated molecular metropolis, operating with a level of precision that is frankly breathtaking. And that brings us to this final thought. You don't just have one of these microscopic cities inside you. You have roughly 30 trillion of them, all working together in complete harmonious synchronization. So, if just one of your 30 trillion cells operates as a highly advanced automated metropolis, how does this invisible machinery dictate our very existence? The sheer scale and brilliance of your molecular architecture is extraordinary, and the more we understand it, the more we understand ourselves. Thank you for joining me on this explainer, and keep questioning the microscopic universe inside you.